Welcome to our webinar on SIBO, or small intestinal bacterial overgrowth, which is currently a popular topic and something we get many questions on. Considering that at longevity, we analyze stool samples, you may wonder what role we play in a microbiome from a different location, that of the proximal small intestine. And that's a legitimate question. With that in mind, let's take a look at what the research is telling us about SIBO, and please note the disclaimer, which is here and on the last slide. Our first reference lists to the left two very important considerations in SIBO. First, it highlights HCl output from within the stomach and how acid blockers can diminish its abundance. Acid blockers may be great for alleviating heartburn when you've eaten things you probably shouldn't have, but they are not good for the long-term health of your microbiome. Keep in mind, you have a microbiome throughout the whole length of your GI tract, not just in the colon. You have an oral microbiota, one in the stomach, believe it or not, in the duodenum, etc., all the way to the very end. The microbiota are not the same from one location to another, as factors such as oxygen availability, motility, stomach acid, luminal pH, bile salts, enzymes, and substrates are variable in different locations. In chronic use of acid blockers, you've not only altered the environment of the stomach, but also the proximal small intestine and what makes its way into the colon, which consists of a high proportion of animal proteins, something that is not good for the colon. So as you can see, the entire microbiome is impacted. But in regards to the stomach, since you've changed the environment, bacteria and fungi who otherwise should not be residing there can now survive. This is about one aspect of SIBO. Another is what they refer to on the bottom left, that of motility, or how quickly the food contents move through the GI tract. Food is supposed to transit the small intestine significantly faster than the colon. When we impact this motility, again, we've altered the environmental basics and allow a shift in who can reside where. To the right is shown 4 trials using antibiotic, rifaximin, along with other agents in the treatment of these bugs where they don't belong. As you can see, the success rate is relatively high. This is the go-to pharmacological approach to SIBO, but doesn't always work, hence the addition of the other agents. Here we'll revisit one of our previous concepts, that of transit time. I know this slide is a bit busy with acronyms, but what these researchers are saying is that those with SIBO have significantly longer GI transit time, specifically for the small intestine. The longer it takes for food to pass through the small intestine, and the more bacteria which accumulate there, the more fermentation you'll have, and therefore more gas and bloating in a place where it shouldn't be. This is a classical symptom of SIBO and something we aim to resolve as part of an overall approach. In this paper, once again, we see our two concepts, HCl suppression and motility. If you look at table 2, you'll see highlighted that significantly more subjects who tested positive for SIBO were on acid-suppressing PPIs as compared to their counterparts who are on a PPI plus a prokinetic. A prokinetic is something that speeds up that motility. One point to take away is that even with PPI use, increased motility can improve outcomes on its own. As for the left side, I'm going to read the text verbatim as it's quite powerful. The authors from this 2018 paper state, chronic acid suppression with long-term PPI leads to hypochlorhydria, which is low stomach acid, which alters the intraluminal environment to facilitate the growth of the bacterial flora in the small intestine and contributes to the proximal migration of colonic microflora. That last bit, proximal migration of colonic microflora, means that bugs that are dwelling further down your GI tract in your colon are able to migrate or work their way up the GI tract to reside in places they don't belong, whether it's in type of bug or quantity or both, and this is the result of acid-suppressing agents, namely PPIs. I'll try to summarize this dense slide for you. What they are saying is that when looking at the middle section of the small intestine in SIBO patients, the types of bacteria that were found were the ones listed to the left. we've identified these bacteria as highly problematic in numerous human fecal analyses. In other words, many of the same bad actors from the colon are residing in the small intestine and SIBO, according to these researchers. Not only are they residing there, but they are dominant. The microbiome is a two-way street. Bugs from the upper GI tract can make their way to the lower GI, and the reverse is true. Either way, we can be dealing with a situation of what I call bugs where they don't belong. Ideally, we should have a properly functioning GI tract with more distinct delineations of residence in their proper location. Once again, we see the now familiar theme of dysmotility in PPI use. This time, these researchers looked at the duodenum, the first part of the small intestine just after the stomach. They found several of the same bad-acting bacteria identified from our previous slide, as well as another topic we get many questions on, that of Candida. Here they use the term SIFO, or small intestinal fungal overgrowth, and Candida was the culprit here, all the way up in the duodenum. Again, here we have bugs residing where they don't belong, thanks to environmental shifts centered around PPI use and dysmotility, both of which were found to be risk factors for overgrowth. Here we have a slightly different take on a now familiar theme. Once again, the researchers looked at the duodenum and found that there was significantly more bacterial overgrowth in the duodenum of the SIBO subjects versus the non-SIBO ones. In addition, we'll now introduce the connection of SIBO to IBS. Here they show that those with IBS had significantly higher levels of duodenal overgrowth as compared to non-IBS. So just a refresher, IBS is something we associate with dysbiosis further down the GI tract in the ileum and colon. But here we're looking way up next to the stomach in the opposite direction. If we put this together with the bad bugs from previous slides and the knowledge that those same bad bugs drive dysbiosis in the lower gut, we see the smoking guns of the same bad actors which can cause problems in one location and can migrate under environmental changes to cause problems in another. This is a continuation of our previous slide. Here we join the two concepts of IBS and PPI use in regards to SIBO. Although they sound like separate concepts, they are highly correlated. Remember, PPIs change the entire microbiome, and the bad actors in IBS can migrate up the GI tract and potentially cause a condition we identify as SIBO. But we can't think about the GI tract as different segments or even different diagnoses. Bad actors, or more accurately, opportunistic pathogens, are bad for any condition. It just depends on a variety of factors. But it's this same large group of opportunistic pathogens which have been strongly associated in many human trials with a wide variety of conditions, ranging from Crohn's disease, ulcerative colitis, and IBS, to depression, anxiety, metabolic syndrome, dementia, and more. Here, we see that SIBO was found in 60% of IBS diarrhea sufferers, and that, once again, PPIs were linked to SIBO. In addition, all of those with IBS had more than a 6-fold risk for also having SIBO. This is something that makes sense as we now understand the bidirectional effects of the GI tract. In this duodenal aspirate study from 2019, I want to hit on some new points. One, it's the symptoms which are the main driver. In other words, SIBO testing is not fail-safe. Just because you're told you haven't tested positive for SIBO, that doesn't mean you don't have it. Although this is aspirate, and again, in-office testing is via breath, the same concept remains. As you can see in the middle of figure A, many symptomatic patients came back as SIBO negative. Point 2, in those with symptoms, the genus Prevotella was low. We've identified the genus Prevotella as a key player in gut health, particularly in diarrhea, which, if you recall, was highly correlated to SIBO in a previous slide. The last point here is that of simple sugars. As expected, they triggered symptoms for these patients, and this is not a surprise. As we now know, there is an excess of bacterial fermentation in the upper small intestine in SIBO. And if you give these bugs a favorite fuel, that of simple sugars, then they'll do what they do best. Of course, a part of any SIBO protocol should be to limit simple sugar consumption. To take our point a step further, if you recall, when a high fiber diet was shifted to a low fiber, and included high simple sugars, symptoms got worse for SIBO. Well, here, the opposite was the case. Again, you see the use of rifaximin, an antibiotic designed to stay within the GI tract, to reduce those extra fermenters who work their way up. Along with a prebiotic, we often recommend partially hydrolyzed guar gum. Here, fiber was added, and with success, as eradication of SIBO was more effective in the rifaximin plus partial hydrolyzed guar group than rifaximin alone. Prebiotics are the cornerstone of what we do. Lastly, I want to touch on a couple new points. If you're familiar with my webinar series, you may recall that the microbiota produces thousands of enzymes. Well, some of those enzymes change the structure of bile salts. Now, we're not going to dive deep into bile, but know that it's released to primarily digest fats and has hormonal implications. In a properly functioning gut, the correct microbiota resides in the small intestine and all goes according to plan. But in SIBO, these extra bugs reside too far up the GI tract, and some of them are now able to perform their enzymatic action on bile salts, changing the structure and function such that it reduces fat absorption. This has multiple effects, one of which is similar to PPI use, in that we now have extra fat making its way into the colon. Although bad bugs tend to like animal protein, fat is also a substrate that can make them happy. Another point from this paper is that of ileal breaking. When these macronutrients make their way down to the distal small intestine, the body reads this as too much food escaping digestion and slows down motility. If we recall from our first slides, motility plays a huge role in SIBO. So as you can see, it's all connected. The overall proper functioning of your entire GI tract is tied into SIBO, something generally considered isolated to the proximal small intestine from a limited point of view. And since we can identify dysbiosis from your stool analysis, and we have the clinical expertise in gut conditions to include SIBO, which includes some simple things like food choices, what we do is relevant for the SIBO sufferer. In review, we touched on one dietary consideration, gut transit time, proper digestion, and large quantities of bad bugs migrating to places where they don't belong. Therefore, if you or a loved one suffers from SIBO or any gut condition, as well as conditions outside the gut, visit our website at longevity.com. There you can find more educational material for what concerns you, and you'll be able to order up one of our kits and begin the process of feeling better finally.